Well, thank you very much for that introduction and the invitation to participate in this very interesting and exciting series. I think you guys have heard now 4 lectures about cancer, so you have a pretty good sense of some of the exciting progress that's being made in the cell biology and the genetics of cancer. And I know that Hal Burstein talked to you recently as well about some of the new drug approaches to cancer based upon the improved understanding of the mechanisms underlying cancer. So to sort of round out the program, I'm going to talk to you about the host response to cancer and introduce you to some of the current thinking in immunology and how the relationship between the tumors and the host is actually quite complicated with divergent roles for the immune system in either inhibiting tumor growth or promoting tumor growth. So I understand this can be very informal, and please feel free to ask questions. It's really a privilege to talk to high school science teachers. And actually, I was recently contacted by a high school, a group of 9th grade students from Bergen High School in New Jersey who are participating in, I think it's a fascinating program of generating their own websites in either science or engineering. And this is a program that's sponsored out of the University of Florida. I'm not sure whether any of you know about this, but it was actually quite sophisticated when they contacted me in terms of the project that they had done last year, which was to make a website on gene therapy for Down syndrome. So I think it's absolutely fantastic now with the internet, how high school students are able to interact with folks in medical schools and so forth. And it's I think crucial that the excitement of research and the great opportunities for improving healthcare are taught to students at the age where you all are focused. So. The immune system can be broadly divided into 2 major arms. One arm, illustrated in the left circle here, is the innate immune response. And this is the response that's rapidly triggered following a microbial infection. And it functions to achieve initial control of infection so that most of the microbes that we're exposed to all the time you don't even know about in terms of disease because this part of the immune system is so efficient. The innate response is composed of a variety of cells. I won't go into the details about these, but you can see that there's a diversity of cell types involved. And there are also soluble factors that circulate in the blood, such as the complement system. Now, in contrast to the innate response, which uses germline encoded pattern recognition receptors to detect the presence of microbes, the adaptive immune response is much slower to develop. And this is because the adaptive immune response manifests exquisite specificity. And this is due to the somatic rearrangement of genes that encode either immunoglobulin molecules or antibodies, which are the specific recognition element of the B cell and the humoral arm of the immune system, or that give rise to the T cells, which recognize actually processed peptides, which are derived from any intracellular protein. These cells, with the exquisite specificity conferred by these 2 receptors, are very rare in the repertoire of a resting host. And that's why, in order for them to get expanded to any significant number, it takes time. But these cells are absolutely crucial as well for controlling infections, because if you lose the function of them by virtue of some inherited immunodeficiency, or now most commonly as a consequence of infection with the HIV virus, patients succumb to a whole variety of infectious diseases. Now, the adaptive immune response is also essential to the development of memory. So it has the unique capacity to actually recall previous encounters. And this memory can last for the lifetime of the host. And the development of memory is actually the basis for why you can develop vaccines, with the concept being an initial exposure to the agent that you want to immunize against is going to be remembered for a very long time. And then when the host encounters the real thing, the host will be prepared by virtue of both B and T cells. And now there's extensive interactions and cross-talk between the innate and adaptive immune response. And there are actually also some cells which share features of both. Now, with that as a background for some of the players in the immune system, it turns out that there are several different possible roles for the host response to tumors. And we'll touch upon today evidence that the host response may actually be involved in preventing tumors. Alternatively, the host response under other circumstances can actually contribute to tumor development. And then regardless of these responses, which occur naturally during the course of tumor development, it's possible to potentially manipulate the immune system in specific ways to develop an anti-tumor immune response that's capable of leading to tumor destruction. So how might the immune system be involved in preventing tumors? Perhaps the clearest evidence for a role relates to the well-known involvement of the immune system in controlling the response to infectious agents. And it's clear that although there are many causes of cancer, including carcinogens, of smoking, alcohol, etc., it's also clear that infectious agents contribute currently to at least 15% of all tumors worldwide. So this is a very, very substantial contribution. And it's likely, as tools for discovering new microbes become more sophisticated, that this number of infectious agents will continue to grow. Now, we currently understand a role for infectious agents in cancer in two main ways. One is where the agent directly contributes to cellular transformation. And examples of this, I apologize for the use of abbreviations, HPV, which is the human papillomavirus, which I'll talk about in more detail in just a second, and Epstein-Barr virus. These viruses have gene products in them that when they are introduced into infected cells, alter the processes that undoubtedly you've heard about in the earlier talks that regulate cell cycle progression, cell death, cell differentiation. So in these cases, microbes directly cause cancer. But there's a large group of agents that don't have transforming genes contained in their genome. And instead, these agents cause inflammation. And because the host is not able to eliminate these pathogens, there's ongoing inflammation. And the setting of ongoing inflammation creates the milieu where cancer can develop because there's a lot of cell turnover. There's mutations that are being introduced into the target cell DNA by virtue of the immune response to these organisms. So some of the innate immune cells, the granulocytes, are able to produce reactive oxygen species, which actually can be mutagenic. So some of these agents include the Helicobacter pylori bacterium, which was only recently learned to be the cause of ulcer disease, gastric ulcer disease. But it infects an enormous proportion of the world's population, and it causes stomach cancer, which worldwide is the second leading cause of cancer death. HBV and HCV refer to hepatitis viruses, either hepatitis B or hepatitis C, and we now know there are many different kinds of hepatitis viruses. These are major causes of liver cancer, particularly in Asia. Now, the importance of identifying pathogens that are linked to cancer is illustrated in a very dramatic way by a study that just appeared in one of the leading clinical journals, the New England Journal of Medicine, last fall or last winter. And this trial, which I'll walk you through, actually represents about 2 decades of work. And it's the culmination of tremendous collaborations between virologists, epidemiologists, and clinicians. And it's been delineated that although there are many different kinds of human papillomaviruses, a few of them are most tightly linked with a risk for cancer. And in this case, the major cancer associated with this virus is cervical cancer, which is also a leading cause of death in women worldwide. Now, what was learned is that there is an immune response to papillomavirus. And this happens after a patient is exposed to this virus, which is transmitted sexually. And sometimes that immune response is able to clear the infection. But in some cases, the virus persists despite the infection. And when the virus hangs out for a long time, that's when it ultimately, over time, can give rise to cancer. So the question was, could you develop an immune response to the papillomavirus before infection and then be prevented from the consequences of infection? So using now basic molecular biology techniques, it's possible to express just a limited number of the gene products that the papillomavirus normally has. And when you express these gene products in a permissive cell, they actually assemble into viral particles that very much resemble the real papillomavirus, except they're devoid of all the nasty genes that are capable of causing cancer down the road. But from the immune system's point of view, exposure to these particles results in a specific immune response. And so, I don't know, I hope this can project, but if not, I just wanted to present to you actually some primary data from this study. So what was done in this study was to identify either late teenagers or women in their early 20s who had no evidence for a papillomavirus infection but were sexually active. And then this study, which involved about 2,500 women, there was a randomization to receive a vaccine composed of those viral particles that I just showed you, or a placebo control. And what was remarkable is that this vaccine was 100% protective to the thousands of women who received it. All of the patients who received the vaccine developed specific antibodies that had the capability of preventing papillomavirus from establishing infection. And in fact, all of the immunized women were completely protected against developing HPV, even though they remained sexually active as before. Whereas the unimmunized women developed papillomavirus infection at the rate that's well established. So this is really a landmark study because it tells us that if we can mass immunize both men and women prior to reaching the age of sexual maturity, we should be able to prevent papillomavirus infection in the forthcoming generations. And as a result of preventing this infection, we will be able to prevent cervical cancer. Turns out there are a couple of other types of papillomavirus which are also in play, and vaccines will need to be prepared against them. But the principles illustrated in this study should be readily transferable to those papilloma subtypes as well. And this kind of study also follows up similar work that's been done with the hepatitis B virus, where immunization programs were started in the Far East now about 2 decades ago, mass immunizations in Taiwan, mainland China. And the incidence of hepatitis B virus has been markedly reduced in the Far East. And 10 to 15 years later, it's now clear that the incidence of liver cancer is also dramatically being reduced. So this inspires a lot more work to try to identify additional pathogens that are linked to tumors because prophylactic vaccines can be highly efficacious. Now, what might be some of the immune mechanisms involved in preventing tumor formation and involved in the response to infection? And here I just want to tell you a little bit about some of the studies that we've done focusing on cytokines. Cytokines are secreted or membrane proteins, which are crucial for regulating the function of the immune system or the blood system. And these proteins are produced by cells and either function back on the cell that produces it, so-called autocrine fashion, or affect cells in the nearby vicinity in a paracrine way. So we've ended up making genetically engineered mice that lack several of the important cytokines involved in the host response to infections. And I won't go into the details of what these cytokines are, except to illustrate to you that the immune system really is combinatorial, and that these cytokines function in collaboration with one another. So that if you make animals that are unable to produce combinations of these cytokines, you start to see dramatic defects compared to animals which only miss individual of these cytokines. And in particular, we find that animals that lack the combination of these cytokines survive very poorly, and about half of them actually die during the first months of life. And that in contrast to their control animals here, they also start to fall off at about a year of age, which corresponds to middle age in humans. And what's going wrong in these animals that lack cytokines is that they're developing infections in part. So the animals that die in their early phase of life die because they develop pneumonia, which is due to bacteria which normally live in the airways of mice but are not causing any kind of harm because that innate immune response is keeping them under control. But when you cripple the immune system, then the bacteria are able to grow and, in this case, fill up an entire lung of the animal, which is why the animals don't survive. But interestingly, the animals that do overcome this initial infection, as they age, they develop cancer. And the prevalence of cancer in these animals is quite high, approaching 80% of the mice. And they develop lymphomas, which are malignancies of lymphocytes, those B cells that I mentioned at the beginning. And they also develop a variety of tumors in other solid organs, like in the ovary, but actually a fairly diverse range of tumors. And in many ways, these mice look a lot like AIDS patients who develop infection and then can develop tumors secondarily. So we wondered actually in these mice whether infectious agents were directly linked to the tumors, a la what I showed you before with the Helicobacter and the hepatitis stories. Here's just illustrating to you what if a pathologist looked at what these tumors, what a pathologist would see looking at these tumors under the microscope, where there's just uncontrolled proliferation of cells that all look the same in a lymphoma. And this is a type of tumor that arises in the ovary called a choriocarcinoma. So to ask the question whether microbes are causing tumors in these animals, we actually put the animals from birth on an antibiotic called enrofloxacin, which is very similar to maybe an antibiotic that some of you have been on or know people on, ciprofloxacin. And we chose this antibiotic because we knew some of the bacteria that were growing in these animals, and we knew that this antibiotic was able to kill those bacteria. So we put a large number of animals on these antibiotics from birth. And the really interesting finding was that these antibiotics completely prevented all of the tumors from forming in these animals, which clearly said that specific kinds of immune deficiency give rise to cancer because of an inability to control infection. Now, we also noticed in these animals that not only was their infection controlled, but they had a marked decrease in inflammation. And we know that inflammation that's uncontrolled in the human is linked to cancer in a very strong way. For example, smokers are at very high risk for lung cancer. And the lung cancer arises in chronic inflammatory disease in the airways. And it's likely there's this synergistic play between the inflammation as well as the carcinogens that are in tobacco smoke that give rise to cancer. So we wondered whether, in fact, apart from the infection, maybe inflammation was contributing to tumors. So to test that idea, we actually put mice on anti-inflammatory drugs. And these are like the Motrins and the ibuprofens that you may be familiar with. It turns out one that's very useful for studies in mice is Sulindac. And Sulindac has actually been used, given to patients quite a bit as well. And what was interesting is that this anti-inflammatory agent, which doesn't have any effects on microbes, also markedly reduced tumor development in these animals. Not quite as potent as the antibiotics, but nearly so. So this is telling us that inflammation is causative for cancer as well in this model. And this fits now with a lot of clinical data that have shown that anti-inflammatory drugs are associated with reduced incidence of cancer. In this study that I'm illustrating here, this is an analysis of a large cohort of women who've been followed for decades, and in which there's been very well documented the development of whatever pathologies happen. And because there's a lot of data collected about lifestyle, potential risk factors for cancer, it's possible to go back and try to draw relationships between particular risk factors and cancer. And it turns out in this study that the risk of breast cancer is reduced in proportion to the amount of nonsteroidal anti-inflammatory drugs that the women were taking. And they would have been taking these agents for musculoskeletal complaints primarily. But it's interesting that there's a pretty strong relationship. And it's been shown similarly, in this case in a prospective way, that patients who are at high risk for developing colon cancer, if they take these same sort of anti-inflammatory drugs, you can markedly reduce the development of colon cancer.
Would aspirin be an anti-inflammatory?
Aspirin is. It turns out with aspirin, the dose is very important. And there's still some controversy about what the optimal dose is. And I think actually one of the hardest decisions in clinical medicine today for me is whether everybody should be taking aspirin or a variant of it. Because there's pretty compelling data across many tumor types that aspirin or Sulindac can prevent tumors. And there's also pretty good data that it can reduce the risk of cardiovascular events. But on the other hand, there's a real risk of intracranial bleeding, stroke, and bleeding internally in the gastrointestinal tract. So it's very hard to sort out right now the relative risk-benefit ratio, because you're talking about taking these drugs over a long period of time, and it's difficult to do a study. But as more evidence comes out in terms of what the specific inflammatory mechanisms are, it may be possible to fine-tune who needs anti-inflammatory agents. And so to tip the balance, the risk-benefit balance, more in favor of, well, the likelihood of your deriving benefit in terms of reducing cancer outweighs the risk for bleeding. But that's still an area that needs more study. So I think this part of the talk has shown that tumor development, both in animals and in humans, can be attributed to an interplay of infectious agents and immune responses which are incapable of completely eradicating particular microbes. And that one of the ways that inflammation in cancer is linked is through specific defects in the cytokines that we've shown. But it's likely that there will be multiple ways that immune deficiency links inflammation in cancer. And this says that it's complicated in terms of preventing cancer. On the one hand, we want to use immunostimulatory approaches like vaccines to make the response to infectious agents better so that they don't set up a nidus for chronic inflammation. But on the other hand, if you do have chronic inflammation, you want to use manipulations that reduce the amount of inflammation, anti-inflammatory strategies. So we need to be a lot smarter in terms of targeting those two different types of intervention to the right group of patients. And I just want to acknowledge some of the folks in my lab who contributed to the work on the cytokine deficiencies. Now, what about an immune response to already established tumors? Is the immune response important in either inhibiting or accelerating tumor growth? And one of the very provocative observations also was published quite recently in January of this year from a group at the University of Pennsylvania, where they studied women who have ovarian cancer. This is actually the most lethal form of cancer in women. And what these investigators were interested in was to examine the nature of the immune response at the time patients were diagnosed with their cancer, and then to see whether the presence or absence of particular immune response was linked to how well the women did. And what the investigators found was that women who showed evidence of an immune response in their tumors, so here the blue background are the ovarian cancer cells, and the brown staining here is actually a particular kind of the T cell. So it turns out in a proportion of women, there is evidence that T cells are recognizing their tumor. Now these T cells by themselves are obviously not doing a whole lot of good because the woman has developed a potentially lethal disease. But what's quite interesting is that when these women then have the standard treatment that we have today, which is surgery to remove the tumor, and then several months of chemotherapy or drugs that basically induce DNA damage, and that cancer cells are less effective at repairing the DNA damage, and therefore there's some selectivity for killing cancer cells over normal cells. Although chemotherapy, as probably Hal talked about, has a lot of side effects because the selectivity is not that great. But what's fascinating here is that the women who showed evidence of a pre-existing immune response, these are the women who were cured by the standard treatment, whereas the women who didn't have any evidence of an immune response all succumbed to their disease. So this is providing a hint that maybe an immune response against the tumor can be integrated with other forms of cancer therapy to result in a synergistic anti-cancer effect. And that if you don't have an underlying immune response, perhaps cancer treatments won't work that well. So this sets the question for investigators in the field of cancer immunology to devise ways to convert patients who don't have immune responses to their tumor to patients that do. Now, there's been a lot of progress in this field in the past 10 years. in terms of devising rational ways to accomplish this goal. And it turns out that genetics and cell biology have been very helpful in leading to the development of methods where you can actually characterize at the molecular level specific targets in cancer cells that the immune system can recognize. Knowing these targets has then allowed a more careful characterization of the spontaneous immune response to tumors. And it's become clearer now that there are reactions to tumors in many cases, but almost always they're weak. They're not the magnitude that you'd have if you were exposed to the flu, for example. But a lot of work has led to an understanding of how do you convert these weak responses into more potent ones. So using that knowledge, it's been possible to devise several different therapeutic approaches to improve the immune response against cancer. And these can be divided into two broad categories. One, passive immunotherapy. This means that actually you're going to instill into the patient some pre-existing component of immunity, which has been derived from some other source. And the two categories that we'll touch on are the use of monoclonal antibodies and bone marrow transplantation. The other approach is to follow up the story that we talked about with the papillomavirus. And this is to actively immunize patients, in this case against their cancer cells, to try to stimulate their own immune responses against these tumors in a way that doesn't happen as part of the normal development of cancer. Now, monoclonal antibodies are quite an interesting story in terms of how good ideas in science sometimes take a long time before they actually reach clinical fruition. The technology underlying monoclonal antibodies actually was awarded a Nobel Prize and developed in 1975. And at this point, it was anticipated that these might be used as magic bullets. Antibodies that are going to target cancer cells specifically and be able to eradicate them. But it turned out that those initial ideas were far too simplified, and that it took a long time to actually figure out how to use these antibodies well, figuring out the best targets, the way these antibodies worked. Turned out initially these antibodies were made with mice, but it proved important to make humanized antibodies. And then it's actually also been shown that you can make antibodies more potent by chemically coupling them to other kinds of killing strategies. But as of today now, 11 of these antibodies are already FDA approved for therapeutic use. These have been shown clearly to prolong survival for patients with diverse hematologic tumors like lymphoma and leukemia, and also women with breast cancer. And the area is just incredibly exciting with currently 400 other antibodies that are in active clinical testing. So it illustrates that there was a lot of clinical investigation that had to be done in order to figure out how to use these antibodies properly. Now, bone marrow transplantation has a similar history, originating in concept also about the same time as monoclonal antibodies. And in this approach, sometimes, and most commonly, cells are taken from an immune-related donor who's healthy, and then infused into a cancer patient. And this is called allogeneic bone marrow transplantation. In some circumstances, actually, the bone marrow from a patient is removed prior to delivering high doses of chemotherapy and radiation therapy, which can be effective for killing cancer cells, but also kill the normal bone marrow cells. So by having harvested bone marrow prior to the administration of those toxic agents, this bone marrow can then be reinfused into the patient and reconstitute their blood system. And it's clear that bone marrow transplantation is very effective therapy for many hematologic tumors and has led to the cures of lots of people. This is a little cartoon which illustrates that the donor here, the bone marrow, can be removed typically from the pelvis, although now there are also techniques where blood cells are obtained from the peripheral blood. And those bone marrow cells have all those immune cells that I showed at the very beginning. And these then are infused into the patient. And then these healthy cells can fight the cancer and lead to an important anti-tumor effect. Now, what about active immunotherapy? So it turns out over about 15 years of work, the mechanisms that how the immune system comes to recognize cancer have been delineated. And I'll point out a couple of things because they'll be relevant in the later part of the talk. That this is where this cytokine GM-CSF that we made the animals that lack this come into play. Because this cytokine is very good at stimulating a specialized immune cell called the dendritic cell. And the dendritic cell is what we call a professional antigen-presenting cell. And what that means is that there's a part of the immune system which is devoted to jump-starting the whole reaction. And the key to that step is the dendritic cell. And GM-CSF is key to making the dendritic cell do its job better. As a result of activating these dendritic cells then present tumor antigens in a very favorable context and are able to stimulate now T cells and also B cells, which are now able to recognize tumors and to attack them. So with that kind of cartoon, you can imagine that there are many permutations of vaccination strategies that need to be tested in humans in order to sort out which are the most effective. And here, some of the issues in terms of what do you immunize with are whether you use whole cancer cells, because they present a very large repertoire of targets. And the use of these whole tumor cells doesn't require definition of specific molecules, because that's still a tricky area. On the other hand, coming up with target antigens that are defined is much more amenable for widespread testing, because you can make vaccines from defined products as opposed to using whole tumor cells, which might be more custom made for each patient. And then there's a variety of strategies which are under testing, all aimed at improving the function of those dendritic cells, getting them activated in the right way and getting them loaded with the right material. So we've been developing an approach now that we've been working on for about the last 10 years or so, in which we're actually going to focus on using the whole tumor cell and use gene transfer technologies to make these tumor cells more immunostimulatory. And in experiments that were started when I was at the Whitehead, we found that this GM-CSF molecule was the best one for introducing into tumors, in animals at least, for then stimulating this immune response. And when I went over to Dana-Farber in '94, we initiated a clinical program to try to test this concept. And we've made significant progress during this time, and actually now have active vaccine programs in many different kinds of cancers in patients. And the basic idea with the approach is that initially these studies need to be performed on patients with advanced disease, because it's introducing a new technology, safety profile is unknown, efficacy is unknown. And so essentially patients for which there is no effective known therapy become candidates for these early phase trials. And necessarily then, these are patients who have advanced disease with tumors in multiple locations. So these patients undergo a surgical procedure to remove some of the tumor. You can't remove all of it at this point in the disease. And then we use different replication defective viruses, or viruses that we've engineered so that they can introduce this GM-CSF gene into the tumor cells very efficiently, and now confer the ability to make GM-CSF to tumor cells which otherwise don't have that property. Then the cells are irradiated. That basically prevents them from growing, because we're going to be giving these cells back to patients, and irradiation provides an important safeguard so that these cells can't become more virulent when they're introduced. But the irradiation doesn't kill the cells. They remain alive and able to produce the GM-CSF hormone for several days. So then these cells are administered into the skin of patients, much like a flu shot. And what we find is that these injections do cause a local inflammatory reaction that looks a lot like a mosquito bite. It's a little bit itchy, but otherwise there's really no significant side effects. And when we do a biopsy of the skin and examine what's happening in this red reaction, we see a large number of these dendritic cells, here marked with a specific antibody, accumulating at the site of this tumor injection. And this is something that doesn't happen normally. But this is what happens when you get a flu shot, except instead of it being against the tumor, it's against the flu virus. Now, as a result of that manipulation, you have a local immune response, but just like with a flu vaccine where you get a shot in the arm, but it protects you against an infection that comes in through the lungs, the vaccine here also leads to immune cells that can circulate throughout the body and to recognize tumor cells all over the place. And here's a tumor that we took out after the vaccination and compared it to the one before vaccination. And before vaccination, there is no immune response. But after vaccination, there's a very strong one. And this is just the same kind of response that was present in those ovarian cancer patients who did well with follow-up standard therapy. So we now know that we can accomplish this conversion from no reaction to a good reaction in about two-thirds of the time. And what we're encouraged by is in our most mature program, which is for a skin cancer called melanoma, which unfortunately is increasing in incidence dramatically and will affect about 1 out of every 10 people in their lifetime now. In the most advanced stage, here the stage 4, the average survival is really only on the order of 6 to 8 months. So we're starting to get the sense that a proportion of these patients really are responding to the vaccine quite well and are out now for almost 4 or 5 years. And this clinical trial represents the collaboration among many clinicians and laboratory people over at the Dana-Farber and Mass General and Brigham and Children's Hospital. So on the one hand, we think we're making some progress because about 1/3 of patients are showing these very good responses. But obviously, the majority are suffering from progressive disease and still succumbing. So what can be learned about the factors that are limiting the efficacy of these vaccination strategies? And it's become clearer in the last 10 years that the immune system has hardwired regulatory circuits which normally function to prevent immune responses from going haywire and causing damage to normal tissues, so-called autoimmune disease. And this comes into play in cancer because cancer cells, although they clearly are different from normal cells in many, many ways, they're also partly just like normal cells. And so these irregulatory circuits also dampen the immune response to tumors because they're thinking that the immune system is trying to act against healthy tissue. So it's being tricked. Now, fortunately, it's been possible to devise strategies to interfere with these regulatory circuits. And the idea is to do this in a very careful way, because we don't want to lose all the regulatory circuits, because then the immune system is going to cause severe disease to normal tissues. But if we can do it in a selective way so that the brake on vaccination is released, then maybe the efficacy of vaccines can be enhanced. And we have a wonderful collaboration with a scientist at UC Berkeley, James Allison, who's worked on this approach now for the past 10 years. And it involves now, which is available for clinical testing, a humanized monoclonal antibody to one of these regulatory signals, which is called CTLA-4. So we've started administering this blocking antibody to CTLA-4 to some of the patients who've been on our vaccine trials. And here's an example of patients who have melanoma, which is that lethal skin disease. And what we found is that when you give this antibody to some patients with melanoma, they actually do show some evidence that there is now reactivity to normal tissues. And in particular, melanoma is derived from a specific skin cell called the melanocyte, which normally functions to produce melanin or pigment in the skin, which reduces the toxicity of ultraviolet irradiation. So it turns out when you give this CTLA-4 neutralizing antibody, the normal brakes on immune reactions against the healthy melanocytes is compromised. And as a result, there's an immune attack against the melanocytes. Now, this is actually a pretty mild reaction, and the only consequence is that you get this rash, which then subsides. But importantly, giving this antibody makes the anti-tumor response much more potent. And what we can see is, without going into the details, is just pre and post, there's a big difference in terms of immune infiltrates again. So CTLA-4 is part of the an effective response. And very recently, this is actually data that's just a month old now, we've been trying to optimize the best way of giving the vaccine and then the antibody in combination. And this is data that illustrates a patient who has ovarian cancer. And ovarian cancer produces a hormone, if you will, that is shed and into the circulation. And so it's present in the blood. It's called CA-125. And actually, this hormone is used in screening women for possible diagnosis of ovarian cancer. But it's also used as a monitor of how the cancer is doing. So normally, the CA-125 level should be less than 30. But this is a patient who came on study. As I said, all of these patients have very advanced disease and had failed prior therapies. So this patient had a very high level of CA-125, nearly 2,000. But we made a vaccine from her ovarian cancer cells and administered it for about 4 months. And she developed very strong immune responses in the skin. But these were clearly not sufficient to control her ovarian cancer because it kept on getting worse. But a month after completing the vaccination, we then gave the antibody to CTLA-4. And nothing much happened for about a month. But then the patient developed a rash, and then the CA-125 levels have dramatically fallen. And this is now going out for about a month. And the patient is doing extremely well, and the tumors that can be seen by radiographic studies have markedly shrunk. So this is perhaps a very dramatic example, but teaching us that the immune response can be manipulated selectively and achieve a pretty pronounced anti-tumor effect. And so we now have FDA approval to expand these studies to quite a large number of patients. And we're finding that there's very important interactions between the CTLA-4 antibody and the kind of vaccination that the patients received previously. So this cell-based approach, this GVAX approach, seems to be very synergistic with the CTLA-4 antibody, whereas other kinds of vaccination strategies are less so. And this is teaching us that there really are important differences in the kind of immune response that specific vaccination strategies elicit. So these studies have taught us that regulatory immune mechanisms are very important in cancer patients in limiting tumor immunity. This also tells us that vaccination can give rise to long-lived immune responses, but those immune responses are held in check by these regulatory mechanisms. But nonetheless, appropriate intervention can unleash these memory cells to function more effectively. And this has opened up a lot of interesting questions to figure out what are the best ways to start inhibiting these inhibitors, and to devise other strategies to specifically augment the function of the memory cells. So maybe directly stimulating them as opposed to inhibiting an inhibitor. And again, these studies were a collaboration with clinicians and scientists from many places. So I'll close with a very interesting comment that Paul Ehrlich is attributed with about a century ago. And Paul Ehrlich was the father of modern chemotherapy, immunology, and hematology. And I think that some of his vision is now coming to fruition as a result of significant improvements in understanding the details of the host anti-tumor response, and that the complexity of both positive and negative influences means that combination approaches which tackle each of those individual arms are going to be, are likely to be far more effective than approaches that only target single components. So I'll be happy to take any questions that you may have. Right.
Is that available to be taken by anybody who has antibodies or pap smear, or do you have to be tested to make sure that you have HPV-16?
Right.
And you can take it as a treatment, or is it just given as a prevention?
There are a lot of vaccines targeting papillomavirus in patients who are already infected. Those vaccines are not nearly as effective as preventive ones. And Merck, who licenses this vaccine, is currently devising the large-scale immunization programs. So I don't know whether it's generally available yet, but it's not going to be too long. That vaccine is going to have to optimally be given prior to reaching sexual maturity, because it's possible that you get infected with the first sexual exposure, and then the benefit of immunization is going to be much, much less. So we're right now in a transition period of getting these very exciting results into general practice. And I'm not an expert exactly where that is, but it's going to be. I'm sure it's going to be pretty soon.
But I mean, just considering that usually the virus can become dormant. My understanding is that the virus can be dormant even if you test positive for it. So if you took the vaccine, wouldn't you develop the antibodies?
Yes.
You would be able to.
That's an excellent question, because you're absolutely right that once you've been infected, the virus can become latent and in fact may no longer produce any infectious particles, which in that case the transmission to somebody else is very, very low. But the problem is, because that papillomavirus is a directly transforming virus, that even if it's not producing viral particles, it's infected the cell, and the cell knows that it's there, and it starts to have alterations in the cell growth control network. Now, your question about, well, why, if you then get a vaccine, why isn't it good enough to then eradicate the pathogen? That may depend on some of these immune regulatory circuits, because it's clear trying to use vaccines to treat existing infection, whether it is papillomavirus, HIV, hepatitis virus, is far less effective than preventive vaccines. And it's probably because once the pathogen has taken residence, the immune system has figured, well, it's there, and the trade-off evolutionarily is that we're going to tolerate some level of long-term persistence of microbes so that we don't go damaging normal tissues. But it would be fascinating to see whether if you gave the vaccine with maybe the CTLA-4 antibody, maybe you could potentially eradicate some of these pre-existing infections, and that's under active study now.
What type of cancer is directly transformed from Epstein-Barr virus?
Oh, I'm sorry for not saying that. That's an excellent question. Lymphomas are the most common in the Western world, with Hodgkin's disease and certain kinds of non-Hodgkin lymphoma clearly being caused by Epstein-Barr virus. In the Far East, a tumor which is very rare in the United States and Europe, but very common, is called nasopharyngeal carcinoma. And that's actually because Epstein-Barr virus, its normal life cycle is that it infects epithelial cells in the mouth and throat. And that's why it's transmitted with infectious mono through kissing and other exchange of oral secretions. And then from the epithelial cells, it infects B cells. And then over time, it can transform B cells into the lymphoma. But in the Far East, those epithelial cells can become the direct targets of transformation and then give rise to this nasopharyngeal carcinoma. Absolutely. The question is, how widespread is Epstein-Barr virus? Probably 99% of people in our country, by the time they get to the second decade of life, have infection. So the point is that actually with all pathogens that are related to cancer, it's only a small proportion of the infected patients who actually develop the complications of the pathogen. That 99% of people have an immune system that effectively controls the pathogen. It's likely that the small subgroup who goes on to develop complications have specific immune defects. That as the mechanisms that are involved in the response to pathogens become more clearly defined, it's possible to now go back and start to see what was different about the few percent of patients who get Hodgkin's disease after Epstein-Barr virus versus those that don't. Right now, the mechanisms are still not clear, but there's a lot of promise, I think, for being able to figure that out. And then potentially to specifically augment their responses by giving them whatever is defective.